what you are doing right now is checking that what you wrote matches or is very similar to what I'm about to write. When we're talking about the cell theory, there are 3 points, but they exist in no particular order. So if I ever asked you, what does the cell theory say? I don't care which order you say these points in, as long as you say them all. One of the things that the cell theory says is that cells come from other cells via processes like mitosis or meiosis, cell division processes. So the only way you can get a cell is for another existing living cell to create one. Now, someone asked me earlier, well, what about the first cell? And that is an excellent question. We have some theories on where the first cell may have come from. But for the cells that we have now, they all needed to come from a pre-existing cell. Another point is that living organisms have one or more cells. So if you are a living organism or you were recently alive and you have just died, you had to have at least one cell. Some organisms are unicellular, that means they only have one cell. Some, like us, are multicellular. But living organisms are comprised of at least one cell. And the other point is that cells perform life functions. And another way to say this is that they are the smallest unit of life. So those are the 3 things that the cell theory says. Notice that I did not write the exact words that your textbook wrote, and I would not expect you to either. I would expect you to be able to write what those 3 points are saying, what they mean. And so direct memorization of exactly the wording that you have in that handout is not necessary, but understanding what they mean is. So for that first question, does anybody want to ask anything about what any of those 3 points is referring to? So then question number 2 asks us about spontaneous generation. Now, we said on the daily review that spontaneous generation is when life arises from something that is non-living. You may have used an example. The example in the handout was, if you take sweaty clothes and wheat, you can make a mouse. That was their idea, because if they did that, mice would show up. And now we're asking, how did we refute it? Which means disprove or provide evidence against it. And there were 3 experiments I told you to look at. Ready? The one that Needham and Spallanzani both looked at, and Louis Pasteur. Now, would I expect you to describe in a huge amount of detail all of these experiments? No, that is not the point. The point is that you understand what they were trying to show. So just to remind you, Reddy is the guy who had 2 containers. One of them had a lid, one didn't. The one with the lid didn't have flies on the meat. The one without the lid did. Needham and Spallanzani were both looking at flasks full of soup and whether or not microorganisms would grow with a lid or without a lid. And then Louis Pasteur did a really similar thing. He had a flask, but his flask had this weird S-shaped tube at the beginning. And so in Louis Pasteur's experiment, he didn't actually use a lid per se. The idea was that this bend in the tube was so small that a living organism couldn't get through that bend in the tube. And so if we look at what's common between all of these experiments, all of them were trying to block access to the sample. So in one of them it was a piece of meat, in the other 2 it was some sort of chicken soup broth. But all 3 of them had in common that they would block access to one of the samples to try to prove that if you blocked access, living organisms couldn't grow from nothing. There had to be other cells there. In Reddy's experiment, he was talking about flies. And in both of these, we were talking about bacteria or microorganisms growing. But the key point to all of them was that if we blocked access to the sample, generally nothing new grew. And so it was helping support the idea that cells could not come from anything. They had to come from a pre-existing cell. And if we blocked the sample so no microorganism could get in, there would be no cells to provide us with support for this theory. One thing that I would like to mention in this category is that Needham still thought that spontaneous generation was true. Spallanzani looked at the same set of data and explained it a different way. And so I put them together because they were talking about the same experiment, but they explained it a different way. Would anybody like to ask any questions about any of those three experiments there? So then the third and fourth question, I'll actually answer them together because it's asking, how did microscopes help contribute to the cell theory, and then who were the people involved? One guy that was involved was Brown. Using a microscope, he realized that cells had a nucleus, so that cells had nuclei. He was studying orchids, and he noticed this dark spot in the center of a cell, and he postulated that that might be something that was actually fairly important. Then we had Schleiden and Schwann. Schleiden was studying plant cells with microscopes. Schwann was studying animal cells with microscopes. So all of these guys were looking at cells with a microscope when they came up with their deductions. What these two guys realized was that there were a lot of similarities between plant and animal cells. And so if there were similarities, then maybe all cells had something in common. The last person is this Virchow guy, and he was the one that postulated that cells come from other cells. So the key point here is that all of these guys came to these conclusions or found evidence by using a microscope and looking at cells. And so the cell theory would not exist if we did not have microscope technology to allow us to look at the internal structures of a cell. I'll just pause here for a second. So it seems like everyone has more or less finished their discussions. So I'll just briefly mention a couple of things because a lot of people asked, in particular about this one. I normally say that your experimental control should be a situation where you don't do the manipulated variable. In this thing, the manipulated variable would be the weight of a stone. And so you can't realistically have a stone that has no weight. If you could, I would say, great, make that be your experimental control. So in this situation, what you probably want to do is not change the weight of stones. So you would want to have a bunch of stones of similar weight, because the idea is that we're trying to prove that our manipulated variable is actually doing something. If I take a person and give them a bunch of stones of similar weight and they throw them all different distances, well then that's saying that the weight of the stone has no effect on how far they throw it. And so having a bunch of stones of similar weight to help set up some sort of baseline would be the idea that you're trying to achieve. So sometimes when you can't say, no, I don't have the manipulated variable at all, what you want to do is not change it. Leave things the way they were normally, or have a bunch of examples where the manipulated variable has not changed so that you have something to compare your results to. Were there any other parts on this chart where people were really a little unsure about what they should write, or maybe other things where you want to just check and make sure what you wrote is correct? So then I'm just going to mention number 2 at the bottom because it's asking basically the same thing, but now you have to set up your experiment. I've suggested to a bunch of people that if you did an experiment kind of like the one Reddy did, where you had one compost that was covered and one compost that was not, that would be a good way to determine whether or not those wasps showed up because of spontaneous generation. So if that sounds like what you set up, that would make a really good experiment. The idea is that you have to try to prove whether or not another cell was necessary to make the wasps, or if they could just show up out of nothing, out of decaying food. So I'll talk for a few minutes about what is on your quest, and then I'll put a few more practice calculations, especially field of use stuff, because I know people often need a little bit more with that. For your quest, please bring a calculator, and if you have one, bring a ruler. If you don't, it will not be the end of the world. I have 15 rulers that I will gladly share with you, and there's only one question where you would need it anyways, so we can easily share. But if you happen to have a ruler, bring it with you tomorrow. For the calculator thing, you won't die if you don't have a calculator. The math is not so hard that you couldn't do it in your head, and you need to be able to use a calculator. Apparently you're having a concert back there. So the first thing that would show up on your quest would be stuff about microscope parts and their functions. And you should be able to, from an image, say which part it is that we're talking about or what that part's function is. The other thing about microscope function that I would ask you about is sort of how to set things up. I gave you a handout way at the beginning that had instructions for how to use a microscope, and it had a picture of a microscope on it. And it said things like, plug it in, turn the light on. And then it said things like, first use the coarse adjustment knob, then use the fine adjustment knob. So knowing kind of in which order you should put things on the microscope and adjust knobs to focus would be something that I would want you to know about. There would definitely be a little bit about things that have happened in the past. And so I'm going to jot down a bunch of guys' names because all of them could be pertinent. At the beginning, we talked about Aristotle, we talked about Hooke, and we talked about Van Leeuwenhoek. Then in the spontaneous generation section, Reddy, Needham, and Spallanzani, and Pasteur. And then in cell theory, Brown, Schleiden and Schwann, and Virchow. So these are a bunch of people's names. Now, I'm not going to be very particular about all of the little details about every single person. In fact, most of the things that I would ask you where you have to refer to a person would either be very basic ideas, like, do you know that Robert Hooke is the one who came up with using the word cell? Or I would give you the opportunity to pick and choose what you were going to explain. So for example, if I asked you on a quest or on a test how microscopes or technology or generally experimentation have led to the knowledge that we have now, you don't have to talk about every single one of those guys. You could pick a few of them. Look at how many points the question is out of and base your answer on that. So if I asked you how microscopes, technology, experiments have contributed to our current knowledge, which I totally would ask you as a written question, you don't have to mention them all. You could pick a few. You could say something like, well, first, Aristotle thought, in the time of Aristotle, we didn't even know what a cell was or what it did. We, in fact, thought that cells came from non-living things. Then people like Louis Pasteur did experiments to refute the idea of spontaneous generation. And then a guy like Brown discovered that all cells have a nucleus. So you wouldn't have to necessarily mention every single person in your answer. Does anyone have any questions that they want to ask about any of those points there? So then the next thing that I'll mention are the types of microscopes. There are optical or electron microscopes. So ones you look through with your eye like we have in the lab, or ones that use a stream of electrons. I showed you the advantage or disadvantages of using them. I also showed you two types of electron microscopes, scanning and transmission electron microscopes. Scanning lets you see the outside, transmission lets you see the inside of a cell. And so I could ask you, hey, in this situation, which microscope should I use? And it would all depend on, did I need the thing to be alive, and was I trying to look at the inside or outside of it? Does anyone want to ask anything about types of microscopes or their advantages or anything like that? So the next thing would be the properties of microscopes. Some of them are basic, like inversion or resolution, where there's not really a calculation involved, but just the fact that you know that microscopes invert the image and that resolution is how clear the image is, or whether or not I can distinguish two separate points. Now, other things involved calculation, like magnification, where you take ocular and objective and multiply them together. Others, for example, like field of view, involve a measurement but also a calculation. So for you to know what the field of view is at a particular power on the microscope, you either have to be able to measure it or you have to know the field of view on another power. So to calculate field of view, remember that you will have two magnifications on one side and the fields of view on the other side. And the key thing is that they're opposites. So magnification on low could go here, field of view on low could go there, magnification on medium here, field of view on medium there. You need to set up a ratio with magnifications on one side, field of view on the other. Now, if you'd rather not set up a math equation, but just think about it logically, please remember that as magnification goes up, field of view goes down, and it has to be by the same amount. So if the magnification gets 10 times greater, well, the field of view has to get 10 times smaller. Now, within this particular section and some of the others, it would be important that you know that there are 1,000 micrometers in 1 millimeter, because sometimes you will be given a measurement in one unit and have to convert it to the other. Would anyone like to ask any questions about field of view right now? So other calculations then would be the size of a cell. To estimate the size of the cell, that's where you take the field of view diameter and divide it by the number of cells that could fit across the field of view. The other calculation would be the scale or magnification of your drawing. That would always be the size of the drawing divided by the actual size. And I'd really like to emphasize units there. It's very probable that you would measure your drawing, say, in centimeters or millimeters, but that the actual size of a cell would be given in micrometers. And so it's really important that you make sure that you have the same units when you're calculating the scale. Does anyone have any questions about either of those two things that they'd like to ask? Yes? So what I usually say to people is I think it looks like the letter U with a line on the front. So when I draw it, I pretend like I'm drawing a really fancy cursive U with a long line at the front of it. Does anyone else have any questions that they would like to ask about any of those calculations? So then we would have cell theory. There's the 3 points. You would have to be able to describe what the cell theory says. So aside from just talking about guys and their experiments, you actually have to know this particular theory. And of course, variables and experimental control, which we just worked on. So you can expect that I will give you an imaginary lab scenario and ask you to identify manipulated responding controlled variables, experimental control, etc. Is there anything that anybody would like to ask about variables or experimental controls? All right, so I'm just going to pause here for a second.